We'll move on to our next speaker. We have Dr. Christian Schuerch from the University Hospital Tübingen in Germany, presenting Architecture of the Tumor Microenvironment Predicts Response to Cancer Immunotherapy.
Hi, everybody. Can you hear me?
Yes. Whenever you're ready, you can share your screen with us.
Yes.
Awesome. Take it away.
Okay. Hi, everybody. Thanks to the organizers for inviting me to this special conference. And today I would like to present to you how we use highly multiplexed tissue imaging to try to predict cancer immunotherapy response. We see tumor architecture as a hierarchy and organization of intercellular relationships. And using these new technologies, we have novel access to the tissue to analyze it in novel ways. First, I would like to give you a short introduction into the tumor immune microenvironment before I go into the details of the method that we use. And then I will show you some results in cutaneous T-cell lymphoma before I reach the conclusions. So the tumor microenvironment is a highly complex ecosystem consisting of tumor cells, structural components such as vessels, fibroblasts, and extracellular matrix, but also a broad range of infiltrating immune cell types, including lymphocyte subsets and myeloid cells. And all these cell types communicate with each other in the TME, and they do so by either secreting cytokines or chemokines or other soluble factors, but also by directly contacting each other. We need novel methods to understand the tissue and tumor microenvironment, especially with regards to the spatial context of the cells. And we need to understand the architecture better to understand the function of the TME, but also to predict outcomes and survival of the patients. And most importantly, we want to understand the cell types and how they interact with each other in order to improve immunotherapies for patients. Recently, there have been exciting developments, especially when it comes to spatial analyses of those questions, and we have different methods to do so. And one of those methods is CODEX, which is called co-detection by indexing. It was developed in the Nolan laboratory at Stanford University, and it uses antibodies that are labeled with unique DNA oligonucleotides, so-called barcodes. And we render them visible in the tissue using a complementary fluorophore labeled probe. Using this, we can do more than 50 antigens at the same time. And we usually, in this CODEX workflow, generate antibody panels. Each antibody has a unique DNA barcode. We then stain the tissue with all the antibodies at once. We render up to 3 oligos or antibodies at a time and then do the imaging. We strip them away, and then the next round of imaging is done with the next set of probes. This is all done automatically in a microfluidic system on a computer automated microscope, a fluorescent microscope, here benchtop version in the Kians. And once we get all the data, we then computationally process the data. We create high dimensional hyperstacks, which we use to segment out the single cells and quantify the marker expression per single cell and also its XY position in the tissue. Using this data, we can then do flow cytometry type analysis, such as cell type clustering. We can map the cell types back into the tissue, and we can also do more complex spatial analysis. So now I would like to show you how we use this technology in a rare cancer called cutaneous T-cell lymphoma. Cutaneous T-cell lymphoma, or CTCL, is a skin lymphoma of CD4 T cells that infiltrate the skin. And it usually starts as insidious patches on the skin, and it takes on average about 6 years until a diagnosis can be made, because those patches look like other reactive conditions like eczema or psoriasis. It usually progresses into more infiltrated stages like the plaque stage and finally the tumor stage. And the patient prognosis in tumor stage is really, really poor. So we hypothesized that the composition of the tumor microenvironment could be used to predict outcome and therapy response in those patients. We had great cohort, we had access to tissues from a CTCL trial where the patients were treated with anti-PD-1 immunotherapy. And we had tissues from 7 responders and 7 non-responders, both before and after treatment. From this, we created tissue microarrays. We used CODEX to analyze 55 markers simultaneously, and we also performed RNA sequencing on step sections of those identical cores. Then we finally did integrative data analysis. So this is how the tissue microarray looks like. This is a 7-color overlay here. We had, as I said, 14 patients, 4 biopsies per patient, 2 pre-treatment and 2 post-treatment. And this cohort has been published by our collaborator, Michael Kudadaust, in JCO 2019. And as you can see here, we had a significant difference in overall survival in the responders compared to the non-responders to pembrolizumab therapy. We performed CODEX using a 55 marker panel. As shown here, we had tumor and T cell markers, CD4, CD8, etc., macrophage markers and other myeloid cells, then functional markers, importantly, of course, PD-1 and other checkpoints, and some markers for stromal and epithelial cells. Importantly, using this panel, we were able to identify at the single cell level the tumor cells and the reactive CD4 T cells. Basically, we used those markers as shown here, and it is well known that tumor cells in cutaneous T cell lymphoma, they downregulate CD7. So it's basically lost on the tumor cells, which is very important because then we can differentiate CD4 reactive T cells from the tumor cells. Also, the tumor cell size, which was significantly bigger than the reactive cells. So I'm quickly showing you some raw data. This is 2 tissue microarray cores. On the left hand, we can see a responder post-treatment, and on the right hand, a non-responder post-treatment. And as it goes through the different cycles, you can see the different markers, how they are expressed in the tissue. CODEX is done by fluorescent microscopy, so we have a very good lateral resolution of about 400 nanometers at 20x, and we can go even higher to 40x or 60x. But for this, 20x was sufficient. And another very important advantage of CODEX is that we can do H&E staining after the run, as shown here, and we have a very nice morphological correlation with our fluorescent data. Using CODEX, we then segmented the cells and we clustered the cells, and we ended up with 21 different clusters, of course, with a lot of manual input and overlaying the clusters on the tissue, etc. And as you can see here, we had 3 different lineages, basically. The auxiliary components like epithelium, stroma, then the lymphoid components with tumor cells, CD4, CD8 T cells, regulatory T cells, and the myeloid lineage with macrophages, Langerhans cells, and dendritic cells. We then mapped those clusters back onto the tissue here in a simplified manner, lumping some clusters together. And as we can see here, compared to the fluorescent data and the H&E, we have a very nice correlation of our cluster maps with those images. When we quantified the cell numbers for each cluster, we did not see any significant differences in the 4 groups, meaning responders and non-responders pre and post treatment. And this is shown here in those 4 different colors. And this was confirming the data from the JCO paper 2019, where they also did not see by standard histology any differences in the infiltrate. However, we saw some differences in very rare subtypes of cells, such as activated ICOS positive CD4 T cells, which were more abundant in the responders before and after treatment, and immunosuppressive IDO positive CD8 T cells, which were a little bit more abundant in the non-responders, as well as ICOS positive highly suppressive regulatory T cells. But those cells were very rare, and we reasoned that besides those cells, there are other features, especially spatial features, that could be more important to predict the immunotherapy response. So we created an algorithm to analyze cellular neighborhoods in the tissue, and a cellular neighborhood in this way is a patch of tissue that has in which each cell has a similar surrounding. So we did this algorithm, and as you can see here, we had 10 different cellular neighborhoods which were enriched in different cell types. For example, the epithelium, which was enriched in epithelial cells, keratinocytes, and some intraepithelial tumor cells. And as you can see here, we have a nice correlation of this epithelial neighborhood with the epithelium as shown in H&E. When we do not see any structure here in this infiltrate in the H&E, we can start seeing structure in the cellular neighborhoods as analyzed by this algorithm. For example, we found a neighborhood here shown in purple, which was enriched in tumor cells and dendritic cells, a neighborhood in yellow, which was enriched in tumor cells and CD4 T cells, reactive CD4 T cells, and a regulatory T cell enriched neighborhood, which is shown in pink. So when we did not see any big differences in the cell numbers, in the absolute cell numbers, we started to see differences in how the cells were spatially arranged in the tissue, as shown by the neighborhoods. For example, we saw that an immune activation neighborhood, as we interpreted this, tumor cells and dendritic cells, tumor cells and CD4 T cells enriched, this neighborhood was increased after treatment in the responders, but did not significantly change in the non-responders. The same here. On the contrary, the regulatory T cell enriched neighborhood was much more abundant in the non-responders even before treatment. So this could, for example, be used as a predictive marker. And again, I want to stress here, the numbers of regulatory T cells were not different. It was just how they were spatially arranged in the tissue that was different between those patient groups. Now, to simplify this highly complex analysis, we came up with the idea of a spatial score. So in our idea, we have an effector T cell, a regulatory T cell, and a tumor cell. And if the effector T cell is close by the regulatory T cell, it gets inhibited in the tissue, right? If it's close by the tumor cell, the chance that the effector T cell can kill the tumor cell is much higher, and it is far away from the Treg and it doesn't get inhibited. So we calculated the distances between all those cells, and we made the spatial score, which is dividing distance A by distance B. And if this score is low, this means that the tumor cell, the effector cell is close by the tumor cell, far away from the Treg, and this is a good sign. So we tested this in our samples, and we found that in the responders pre-treatment, we had a much lower spatial score than in the non-responders, especially for CD4 T cells and PD-1 positive CD4 T cells. This was even enhanced after treatment. And interestingly, we did not see any difference in the spatial score when it comes to CD8 T cells. We then validated the score, the spatial score, on a different platform, which is called Vectra. This is a low-plex platform that can do 7 markers, and we created a panel to basically reproduce this finding. And this is shown here. We calculated the distances between the different cells in the tissues. And again, we found the same result here, even a little bit enhanced, because we had a different tissue sections and also a different panel. But again, basically it was the same result that was confirming the spatial score and its prediction. So now what's the mechanism of this? Why are CD4 T cells so important? And there was a high profile paper last year in Cell that showed that actually CD4 T cells can also be cytotoxic. And this has been around for a while, this idea, but it hasn't been very much promoted. So we found this very interesting. We looked into our data, and we actually saw that our PD-1 CD4 T cells, also some of them express granzyme B, which was in our panel. And when we quantified the granzyme B positive cells among all these patients, we saw that in the responders, they increased after treatment, but not in the non-responders. So this could be a mechanism of tumor cell killing in those patients. We also performed cybersort and RNA sequencing, and in the responders, we saw that after treatment, a lot of different molecules were upregulated, especially CXCL13, which is a chemokine that is important for T-cell migration in tumors, not so in the non-responders. And when we did cybersort to identify the source of those of the cytokine, we basically found different cell types. We could identify them using cybersort in our bulk RNA-seq data. And we found that the CXCL13 is mainly coming from the tumor cells, and the tumor cells are upregulating it after treatment with pembro, but only in the responders, not in the non-responders. And we also confirmed this by doing immunohistochemistry for CXCL13. This is a well-known marker for example, angioimmunoblastic T-cell lymphoma. And we could show in our Vectra data that CXCL13 is mainly expressed in tumor cells. So our hypothesis right now is that we have two different tumor microenvironments in the responders and in the non-responders. In the responders, we have a tumor microenvironment that is prone to immune activation, and we can activate it by giving pembrolizumab to the patients, and this results in effector T-cell activation, PD-1/CD4 T-cells that are able to kill tumor cells. In the non-responders, we have an exhausted phenotype, and this is even more so after treatment, where no effect of the therapy or even a detrimental effect of the therapy is happening. And we think we could show that highly multiplexed imaging of the tissue microenvironment and tumor microenvironment allows prediction of tumor progression, patient outcomes, and immunotherapy response. And we can even discover or create hypotheses of molecular mechanisms and cellular mechanisms that lead to tumor control. So with that, I would like to thank all the people involved. This was mainly done in my postdoc lab at the Stanford University. I recently moved to Tübingen to start my own lab, but this work was done at Stanford, and I would like to thank all the people involved, especially Professor Gary Nolan for inviting me to join this group, Darcy Phillips and Magda and Yuri Goltsev, and also the computational grad students, all the funders. And finally, thank you for attention. I'm recruiting people, so if you're interested in my work, please contact me at this email address, and I'm happy to take any questions now. Thank you very much.
Thank you, Dr. Schuerch, for that very beautifully presented talk. We don't have any questions in the chat right now, but if any of the attendees have questions, please feel free to post them. Pam and Dr. do you have questions?
Sure. So, Christian, that was those images are outstanding. I'm sold. So my question to you is that you've obviously shown that there's an exhausted phenotype in the non-responders, and using the cyber imaging, it's very clear. So can you use that kind of imaging to perhaps direct us to how to find those biomarkers that will enable us to make those non-responders responders, perhaps to other, look for other targets?
Yes, that's a very important question, and we are actually working on expanding our panels to do so. So we're now including more checkpoint markers to see, like TIM3 and VISTA and all those markers, to see whether we can find differences in those markers, and also differences in other cell types or spatial scores in other cell types like myeloid cells, macrophages. We know, for example, that macrophages can express PD-1, right? And the blocking PD-1 on macrophages increases their phagocytosis. Same for CD47 and SERP alpha. So we're actually expanding on those antibodies on those markers. It's sometimes very difficult because this is all done retrospectively, right? So those samples are FFPE samples, paraffin samples that were collected a long time ago, and it's much more difficult to establish antibodies for tissue staining in those samples. It's much easier if we have prospective samples that we can collect fresh frozen, right? But we're actually working in this direction, and also the RNA-seq data has some interesting results that I didn't show now. that point towards potential mechanisms as well.
So that was going to be my next question, that would you actually look at the RNA-seq data to identify what will distinguish responders and non-responders beyond what so that you're analyzing that right now.
Yes, there are so many data to analyze still that I couldn't include them, or I cannot draw any premature conclusions right now.
Thank you.
Really amazing talking. Congrats on the new lab as well. I had a few questions actually, and by the way, I hope we can have you back as a keynote in the future years for the conference. One question I had
too. Thanks.
One question in particular was this really neat way of looking at the, I guess, spatial architecture and where Tregs are compared to T-cons and other subsets in the skin. So you mentioned that the Treg numbers are the same, their location is different. I'm wondering specifically in the single cell, if you had a chance to look at transcriptionally, are these, do they look like activated Tregs when the tumor is there? Do they look like tissue Tregs?
So what I can say is that, so we don't have single cell RNA-seq data, right? It's all bulk, but it's inferred. Right. It's like we did CyberSort to infer from the different subsets of cells to infer their expression data. But this is all in silico, so we don't really have single cell RNA-seq data. But what we found is that the ICOS positive regulatory T cells, which are more immunosuppressive, they are actually they are more abundant in the non-responders.
I see. That's very. And how about, because there's also this data coming from Mike Rosenblum's lab at Stanford about the GATA3 positive Tregs in the skin and so on. Did you find anything with these more canonical transcription factors?
Yes, we actually included GATA3 and TBET in the stainings. TBET is a very difficult marker to stain with. GATA3 did work very well. The problem is that the tumor cells are also GATA3 positive. So we excluded this marker to cluster the cells, because otherwise the clustering would mess up, right? But what actually is also really interesting is that sometimes the tumor cells are even regulatory T cells. In one case, it was the case that the tumor cells had a regulatory T cell phenotype with FOXP3 and everything expression. Yes,
that's very interesting.
It's a really complicated issue, because we have the tumor cells that are also CD4 T cells. So it's actually more difficult to analyze this than, for example, in a carcinoma, where it's clear what is tumor and what is immune system.
Very interesting. And one last question, this going back to my personal interest. I'm wondering, I didn't see in your data anything about gamma delta T cells. Are they playing any role at all or are they just grouped together with the other T cells?
We didn't, unfortunately, did not have the gamma delta T cell antibody available yet, but now we have it. So we have a very good antibody against delta and also gamma T cell receptor, but at the time of data acquisition, this wasn't established yet. So I'm really interested in actually looking into those cell subsets, because we're doing some COVID lungs right now, tissue microarrays from autopsies from COVID lungs. And it seems that the gamma delta T cells, they are playing an interesting role there.
Yeah, especially in the skin, for years, they've talked about them having a sort of tumor surveillance role, but there's very little actual data on it. So it would be really cool to see.
Yeah,
thank you for the really wonderful talk.
Thank you.
So one more really quick question in the chat. Someone is asking, what are your insights of LCOS positive Treg to repress CXCR5, CXCL13 axis?
Oh, that's a very difficult question. I honestly, I don't know. I mean, I know that ICOS positive Tregs are more immunosuppressive because they're activated Tregs, but how they affect CXCR5, CXCL13, I really honestly don't know.
Oh, good. Thank you so much for your amazing presentation, really.
Thank you for the invitation.